All right. Welcome, everybody. We are going to be doing something a little bit different. So we're hoping that this will be the start of an annual tradition, and we'll get a chance to see more of what the trainees here are doing in terms of their research. So without further ado, I'll hand it to Dr. Krantz.
It's a great pleasure to introduce Melanie. Melanie did her undergraduate work at Harvey Mudd, and then did her MD PhD work in the combined USC Caltech program, where she did groundbreaking work with Sarcus mesmanian on the mechanisms by which the microbiome is created in mice. And she's moved now to humans, where she's done outstanding work in 3 different realms. And I'm amazed to watch her do such amazing things in 3 different realms. One, she's an outstanding administrator, and she's done transformative things as the chief resident for the research track. She's an outstanding clinician. I've supervised her in mood clinic, and she's really a great scientist too, as you're going to hear. And she's managed to tap some of the best and the brightest mentors, including Dr. London, Dr. Lavretsky, Jonathan Jacobs, not in our department, Jin Cho, and Mike Irwin. So without further ado, nice to hear you talk.
All right. Thank you, Dr. Krantz. Your introduction was perfect, not too long, and then got all the important information, so I appreciate that. And I'm really grateful to the Grand Rounds Committee for inviting me and giving me the opportunity to present my research in this wonderful forum. So I will be talking about the role of gut microbiome in depression. So microbiome, I'm sure everybody in this room has heard about it at least once in various different format. Over the last decade or so, there has been an explosion of growth of microbiome field, and it's been celebrated by prestigious peer-reviewed scientific journals like Science, Nature, and Cell, all the way down to various social media spheres now. You really can't find connection. These are some of the popular media coverage on a link between gut bacteria and mental health. So this hype has been really good for the growth of the field, but there is a clear concern that the hype might be running ahead of the science. So my focus of this talk is really to first provide an overview of what we actually do know about the human microbiome. And then I would like to review the scientific evidence, trying to avoid any kind of hype, but only the facts of what we know about connection between gut microbiome and depression. And lastly, I'd like to talk about some of the results from the pilot study that I was lucky enough to be involved in to look at the link between microbiome and depression treatment response. So first of all, the definition of human microbiome is defined by all microorganisms and their collective genomes residing within or on human body, and that includes all environmentally exposed surfaces, including the oral cavity, GI tract, skin, vaginal tract. There are 10 times more microbial cells within the microbiome than the number of human cells, and there are 100-fold more genes within the microbiome than the human genome. So this knowledge has really extended the definition of what constitutes a human, in that we actually are not alone, and we're probably superorganism that cannot survive without our microbial friends. Functionally, human microbiome plays an important role in various vital homeostatic mechanisms in the body. A lot of them are within the intra-intestinal compartment, but some of them actually go beyond the intestinal compartment to the extra-intestinal compartment. Some of the examples that we are well aware of is protection against pathogens, as the normal flora in the gut prevents opportunistic pathogens from expanding. They also have metabolic and nutritional capacity, as it synthesizes some of the known vitamins, and it can digest carbohydrates and lipids that humans cannot digest. It is also known to be important for development of immune system, most importantly within the mucosal immunity in the gut area. But lastly, most recently, it's been the topic of interest is how the gut microbiome can modulate the central nervous system, brain, and the behavior. Some other thing we know about the normal human microbiome is that the human microbiome composition varies by region. As the graphic, this figure depicts, there's a drastic variability between the different anatomic sites and their microbial composition. And we also know the human gut microbiome changes over life. So at birth, oops, there you go. Before birth, we are sterile. There's no microbiome present. But upon birth, depending on the mode of delivery, either you get exposed to vaginal flora or the skin flora, and then that starts to build up of your microbiome. And then at the time when the solid food is introduced, that also is a time point of microbiome really exponentially increasing in diversity. And during life, there's a clear change in microbiome when there's an antibiotic perturbation or malnutrition. And we also know that in adult microbiome, there's a clear difference between lean individuals and obese individuals. And the microbiome difference seems to mediate the obesity. And there's a bidirectional relationship. The microbiome continues to change with aging. Although I did not put it in this slide, we also know that there's a great amount of inter-individual variation in microbiome composition. And this is important because when you're looking at microbiome data, you have to always keep in mind that some of the variability is due to the inter-individual variabilities. Some are temporal variability, and some are based on factors like diet, nutrition, and health state. Another thing we know is that dysregulation of gut microbiome, which was referred to, the term has been coined recently as dysbiosis, has been associated with various human disease states. On the left is a graphical representation of how normal individual gut flora is different from individuals with inflammatory bowel disease gut flora. And this type of dysregulation of composition has been found in obesity, as I just mentioned. Interestingly, childhood asthma and allergy has been associated with absence of H. pylori during childhood. Pseudomembranous colitis is a well-known example of antibiotic perturbation of the microbiome, resulting in overgrowth of C. diff. And the interesting question is there a dysbiosis associated with neuropsychiatric disturbance, such as depression? So I will segue into the review of the evidence linking gut microbiome and depression. So this is a seminal study that was done at UCLA that really put the concept of gut-brain axis on the map. In this study, they fed probiotics, the fermented milk product with probiotics, to healthy female volunteers for 4 weeks. And then they compared the fMRI brain reactivity at baseline before probiotics and then after 4 weeks of probiotic ingestion. And there was a clear difference in reactivity among the FMPP group compared to placebo group and compared to no intervention group. This difference in reactivity was identified in 3 distinct brain regions: insula, periaqueductal gray, and somatosensory cortex, and they represent the functional network that's associated with emotional, somatosensory, interoceptive processing. So this is a first evidence that really linked the influence, the direct effect of probiotic in the gut, or modulation of gut microbiome, having an impact on the brain function. This is a interventional study, a meta-analysis of how probiotics affect mood symptoms. There aren't that many studies out there. This meta-analysis was from last year. It included 10 studies, and out of those 10, only 3 of them were studies looking at patients with depression, and then the other 7 studies were studying healthy subjects who were given probiotics and then assessed for mood symptoms before and after. As you can see, there's no significant effect of probiotics in the healthy subjects. However, there was a small effect, but significant effect of probiotics in the patients with depression. This is a cross-sectional study of looking at diet, high probiotic food consumption in diet, and its association with depression prevalence. 760 individuals were surveyed using food frequency questionnaire, and then they looked for high probiotic food consumption, such as fermented vegetable and fermented milk product. This was a study done in Korea, so I put a picture of kimchi there. And there was a significant association when looking at the entire sample, but there was a much greater association in men, and there was no association in women, in that in men, high probiotic food consumption was associated with low prevalence of depression. And this is also an important study in the field of microbiome in the study of depression or mental health, in that this is the first large scale population cohort study of microbiome, where they associated different taxa within the microbiome with quality of life score, as well as depression. This was a population of 1,000, Flemish cohort of over 1,000 people in the cohort. And 2 taxa, Faecalibacterium and Coprococcus, were consistently positively associated with high mental score and physical score within the quality of life. The strength of the study is that they also involved a second independent cohort for validation purpose, and then the check marks indicate taxa that were validated by independent cohort. Looking at depression status, they also consistently found depletion of Coprococcus and Deolister in depression, which was also independently validated. This is an cross-sectional study, though, so it does not tell you anything about causality, whether depletion in these 2 taxa caused depression or depression caused dysbiosis. However, this is still takes us closer to understanding the mechanism. Lastly, I'd like to show you the results from a pilot study, and in this study we asked different questions compared that was not asked previously in understanding microbiome and depression. So the first question we asked was, does the gut microbiome mediate response to treatment in depression? In other words, can microbiome predict whether a person has a higher probability of responding to depression treatment or not? Number 2, does the gut microbiome change in response to treatment for depression? And this was asked to arrive at a potential answer for, is there a mechanistic, a novel mechanism of action by antidepressant when it works in alleviating mood symptoms? So this was a parent study of a double-blind, placebo-controlled RCT of levomilnacipran. Its effect on cortical thickness in late life depression was used as a parent study to propose a pilot microbiome study. This was done at Lavresky Group, and my involvement in the study was only the analysis of the microbiome data. So they have collected stool sample from 17 participants, all 60 years or older, with depression, and they were treated with either levomilnacipran or placebo for 12 weeks. The primary outcome of the study was depression score, and response was defined by remission, defined by a HAM-D score of 6 or less. Out of the 17 subjects that were enrolled in the study, 10 were received placebo and 7 received levomilnacipran, but because of the tolerability issue, 3 dropped out from this arm, and only 4 of them completed the levomilnacipran, and 9 completed the placebo. and here's the summary of results. In the levomilnazepam group, 2 of them were responder and 2 of them were non-responder, whereas in placebo, 33% of them were responder and 67% were non-responder. Due to an extremely small sample size, we decided to combine the data from levomilnazepam and placebo and just go ahead and do all the analysis of responder versus non-responder in terms of comparing microbiome composition. So this is the baseline taxonomic profile of responder versus non-responder. Each box represents a genus of bacteria that's present at baseline, and there seems to be some difference between this group and this group. The difference was now represented in a form of beta diversity, which means you can look at each individual. So each dot is represent an individual, and that dot is numerically represented by to represent the actual composition of the total microbiome. So if the 2 dots are close, that means the composition is similar, and then if they're far apart, that means the composition of microbiome is dissimilar. So 5 responders and 8 non-responders were represented based on their composition, and there was a significant difference between non-responder and responder, P of 0.03, when adjusting for sex and treatment. So it pointed to that there is actual baseline microbiome difference between responder and non-responder. And then I used a machine learning algorithm, random forest analysis, to, out of these differences, which of the taxa are actually important and contributing to the classifier. So 10 taxa were identified through this random forest analysis. The accuracy of this classifier predicting response, treatment response, was represented by area under the curve, 0.95, which was pretty good. So these 10 taxa as a classifier had high predictive accuracy for treatment response prediction. So out of these 10, the importance score was also used as a measure of how important each taxa was in predicting. So this score reflects contribution of the taxa to classifier accuracy. So the top 5 taxa were looked at, and I asked what was the actual relative abundance of each taxa at baseline. Interestingly, this UBA 1819, which had the highest importance score, was depleted in responder, whereas the other 4 were enriched in responders. Another question I asked is there a treatment-dependent change in microbiome? So pre versus post-analysis. Red here is post-treatment, and then baseline is pre-treatment. And interestingly, only in responders, but not in non-responders, was a trend towards a change. So it wasn't significant because of the low sample size, but if you can appreciate the directionality of the arrows, they were all confluent. And then I asked which of the taxa were different in the responders, post-treatment versus baseline. And this graph actually tells you the full change of taxa, enriched taxa in post-treatment, and then depleted taxa in post-treatment among the responders. I had to use the cutoff of Q of less than 0.1 instead of 0.05, again due to low sample size. But when I did that, I was able to identify several taxa that were either enriched or depleted in response to treatment. This was not found in the non-responders. So this kind of change in microbiome was only observed in responders. So in conclusion, I'll just go over all the data, not only mine, but the literature that I also showed you. Probiotic treatment has small but significant effect on depression alleviation, and high probiotic consumption is associated with low depression prevalence in males. Dysbiosis associated with depression is characterized by low abundance of coprococcus and dialister. And baseline microbiota can predict treatment response in depression. And Faecalibacterium is positively associated with high quality of life and also predicts treatment response in depression. I kind of breezed over it, but this was one of the taxa that I identified in my prediction analysis. Antidepressant treatment changes microbiome in treatment responders. So these are the findings. Feasibility, so one significance of this study is feasibility of microbiome as a predictor for treatment response in depression, which was not studied previously. Potential for microbiome as a biomarker for depression treatment decision algorithm was a hope that I had. Perhaps that would be a useful way to use microbiome in the treatment algorithm for depression. Perhaps this data can give us potential for microbiome modification through either direct probiotic ingestion, prebiotics, or antibiotics. Whatever way to modify the microbiome to mediate or augment treatment response. Limitation, obviously, very, very small sample size, limiting analysis options, and replication of the study in a larger group is needed. It would be desirable to have functional metabolomic association with treatment response. So with that, I will wrap up as I think I'm running over. Okay, thank you.
While Emily gets ready, it's my great honor to introduce Dr. Emily Wood today for Grand Rounds. I had the pleasure of knowing Emily through the VA Psychosis Clinic last year, but I do want to say that she's both an extremely accomplished researcher as well as clinician. Her research career actually began during undergraduate when she was already studying dopamine and the substantia nigra, which is much more than I can say, at University of Washington. And then she obtained her MD and PhD from Johns Hopkins University as well as the NANDS. Oh yes, she was looking at MR spectroscopy of multiple sclerosis at the time. So she obviously graduated residency from here and is currently one of the research fellows in the Child Fellow track here at UCLA, and she's currently working with Mirella DiPreto, continuing her work in magnetic resonance spectroscopy, this time in autism spectrum disorder. I do want to say some words about Emily as a clinician. It was really actually great to have both her and Melanie in our clinic last year. And it was amazing seeing her compassion, her dedication, and her willingness to go the extra mile for her patients. We have many, many wonderful residents in our program, but Emily was the only resident out of 9 who, even taking into account no-shows, had a greater than 100% booking rate into our clinic schedule. Anyway, it's an honor to introduce Dr. Emily Wood.
So apparently I also need to take the class on how to say no to my patients, but it's hard. So thanks so much for having me here to tell you about my work. I've been working with Shula Green, Mirella DiPreto, and Susan Buchheimer for the last year or so, and I've been able to do some neat things with them, and I wanted to share with you today my work in studying the relationship between GABA concentration, intrinsic brain connectivity, and sensory over-responsivity in youth with autism spectrum disorder. I'm going to talk a little bit about SOR, sensory over-responsivity, the Salience Network very briefly, the excitatory inhibitory hypothesis of autism and why we care about GABA and why we studied it. Briefly, the methods for this, this pilot data, or we're working on getting more in so it can be more than just a pilot, my results, and then some conclusions and clinical implications that I hope will be useful to you guys. So SOR is extremely common in ASD, and you may know that it has become one of the DSM-5 criteria as part of the restricted repetitive behaviors, patterns, and interests part of autism. It's also pretty prevalent in typically developing youth. And what it is a negative reaction to or avoidance of sensory stimuli. What we sort of think of most commonly is the auditory and tactile stimuli, but there are others. And importantly, SOR is impairing for these kids and their families. It's associated with greater impairment, including more severe autism and lower adaptive behavior and daily living skills, more behavioral problems, externalizing and internalizing behaviors, more parenting stress, and it's highly correlated with anxiety and negative emotionality. And a couple other things. SOR is found in other disorders at higher rates than in typically developing. ADHD, anxiety, early life stress or trauma. And we know that autism spectrum disorder is highly comorbid with fear and anxiety disorders, including GAD, PTSD, OCD, social anxiety disorder. And this is something I want you to think about that we'll come back to, which is that a lot of our kids with autism have anxiety disorders. It's very possible that a lot of the people we're seeing in our clinics that we're labeling as primarily having an anxiety disorder might have some autism. It's a spectrum. And how we treat these kids and adults for their anxiety might be related to whether or not they have sensory over-responsivity. So in our lab, we've found that SOR severity, so more impairing SOR, correlates with neural activation in the bilateral amygdala, the right thalamus, the auditory cortex, and the somatosensory cortex. And let's think about what those parts do. The amygdala is part of the limbic system and the Salience Network, which I'll come back to. It's involved in emotional learning and response and memory modulation. The thalamus is a really key part of the sensory system and is in the Salience Network. And it's involved in integrating, relaying, and inhibiting attention to sensory information. And then we know the auditory cortex and somatosensory cortex process sensory input from those respective senses. And so one thing we talk about in fMRI is these intrinsically connected networks. And what that means is that when we look at brains, sometimes during behavior, but also in the resting state, we find that there are parts of the brain that fire together. And we have come to recognize several different networks, and one of them is the Salience Network, which is anchored in the anterior insular cortex and the anterior cingulate cortex. But it also includes firing with the amygdala, the striatum or putamen, the thalamus, and parts of the basal ganglia. And the reason we call it the Salience Network is it seems to be really important for detecting and filtering stimuli and recruiting relevant functional networks to handle those stimuli. In our lab, SOR has been related to some salic network connectivity differences. And what we've seen is that anterior insular cortex connectivity with amygdala and sensory processing regions is greater in kids with SOR, and that the AIC connectivity with social cognition regions is less in kids with high SOR. So this is sort of really implicating, and other work in other labs has done this as well, implicating the Salience Network in autism as it plays a role in directing attention to the most salient interoceptive and extraceptive stimuli for these kids, and they may not be doing it as well as we'd like them to. Some other evidence that we've looked at that brings me to the work I'm doing today is looking at parts of the Salience Network. So we've seen that compared to typically developing youth, autism spectrum participants displayed aberrant modulation of the connectivity between the thalamus and cortex during sensory stimulation. This was in Dr. Green's paper published in 2017. And what we think is that maybe this reduced thalamocortical inhibition in response to sensory stimulation could be leading to a difficulty filtering out or integrating sensory information. Similarly, there was actually, whereas there was less thalamocortical connectivity, there was more thalamo-amygdala connectivity in kids with greater SOR. And it suggests that an increase in amygdala connectivity with a thalamus might be partially responsible for deficits in selective attention, as the amygdala signals the brain to attend to these distracting sensory stimuli when it maybe should be attending to something else. So about the inhibitory and excitatory ratio in GABA. GABA and glutamate are the major inhibitory and excitatory neurotransmitters of the CNS, and there is a lot of it. GABA is present at 35% of synapses in the CNS, and glutamate, I was surprised, is present in 90% of synapses in the central nervous system. And we think of these as being sort of the major inhibitory and excitatory players. And so in autism, there has been an ongoing question about whether or not there's an imbalance in this ratio of inhibition to excitation. Several pieces of evidence have been that seizures are very common in ASD. There's a high overlap with comorbidity of 30% of children with ASD having epilepsy. There's also been some more specific studies looking at, for instance, that there are decreased GABA receptors subunits in autism and less of the enzyme that produces GABA from glutamate, which is GAD in autism. So you can imagine if you have less of GAD, you actually have less GABA and then more glutamate. Rubinstein and Merzenich in 2003 proposed that this inhibitory-excitatory ratio decrease could be related to sort of some of the broader phenotypic things we see in autism. And that, in the way they describe this for sensory issues is that our system really depends on lateral inhibition to fine-tune our sensory system. And throughout development, our cortical mini-columns are altered, could be altered during critical periods if we don't have this fine-tuning. And what I mean by fine-tuning is that, as seen here, if you are pricked by a pencil, you not only will that point in your skin depress, but there'll be nearby areas that are depressed. But you need to feel where the pencil is actually touching you. And lateral inhibition is the mechanism whereby this larger burst of firing from this central area will laterally inhibit the nearby areas and then allow you to fine-tune and really figure out where that is. And this actually seems to be the case in our tonotopic or auditory cortex as well. And if they recognize that if you didn't have a good specific tuning system, that sensory stimuli could really elicit stronger or more noisy or more variable neural responses and maybe make you hypersensitive. And so this is one of the reasons that we're looking at GABA, in addition to some of that other evidence about why the thalamus is important and the amygdala and how these pieces are playing a role. So I looked at 23 kids so far with autism and 13 typically developing. I'm actually really just showing you the autism data today, and I'll come back to TD at the end. This data is looking at autism. And the behavioral measures that I'm looking at with this are just the sensory over-responsivity measures, and the important part there I want you to know is that the higher score means worse SOR or more impairing sensory over-responsivity. I did magnetic resonance spectroscopy or analyzed this data that's from the bilateral thalamus, and we also did resting state fMRI analyses where the bilateral thalamus was used as a seed in a whole brain connectivity analysis. And then we used that thalamic GABA to glutamate ratio from the MRS as a regressor to determine how GABA concentration predicts thalamic connectivity. MR spectroscopy is my specialty, and I would love to talk to you about it in greater detail, but it's probably not that useful for the moment. And what's important about what we did is that we did a special kind of sequence on the MRI scanner that allows us to separate out to some degree these peaks which represent the GABA and glutamate. Really, when I say glutamate, I'm talking about glutamine, and there's a whole discussion about that, but we're going with glutamate today. And that the area, what's important is that the area of these peaks are a measure of metabolite concentration. And this is sort of showing exactly where in the thalamus we're getting this data from. So when I say thalamic GABA to glutamate ratio, it's because I only measured it from that spot in the brain. So what we found is that worse SOR is correlated with the lower thalamic to GABA glutamate ratio. So when you have this less sort of inhibitory tone, it seems to be related to worse SOR. And then we looked at the fMRI data, and in this, where we looked at how the thalamic GABA glutamate ratio correlates with greater positive connectivity of the bilateral thalamus with the right orbital frontal cortex, bilateral amygdala, bilateral putamen, the bilateral cingulate gyrus, and within the thalamus. So what this means is that in the kids that have more GABA to glutamate, they also had more connectivity between the thalamus in each of these locations. And you might have noticed that those locations were some of the one I mentioned when we were talking about the Salience Network. So, conclusions from this data that we, this preliminary analysis, is that lower GABA to glutamate ratio, or maybe having less inhibitory tone, is associated with greater SOR severity and less resting state connectivity between the thalamus and these critical areas that we know to be involved in modulating the response to sensory input. These data are consistent with our prior research from the Green and DePretto and Bockheimer labs that demonstrated a relationship between atypical amygdala-thalamic connectivity, atypical thalamocortical connectivity, and then abnormal sensory processing in autism. And I'd like to talk a little bit about what this might mean for clinicians. So this and other work from the Green and DePretto labs of brain activation patterns in subjects with SOR may contraindicate exposure therapy for SOR-related avoidance. Okay, so maybe not everyone has been through John Piacentini's anxiety clinic, but this is kind of a big deal. So the thing is that when we are looking at kids with anxiety, trauma, all of these things, we're using exposure response prevention therapy. What we think we're kind of doing is helping those kids or adults bring on their frontal executive, maybe parts of the Salience Network, to allow them to inhibit the amygdala properly so that they aren't attending as much to the things that are really anxiety provoking for them, and then eventually are able to function even when they're having those fearful experiences. And what this information about SOR suggests is that in these kids with sensory over-responsivity, the way that their brain habituates to sensory information may actually be different in such a way that we've, for instance, seen that over time when they're exposed to a certain stimulus, their neural response on TDs goes down, ASD keeps going up. And this may be related to these fundamental differences in their Salience Network, which could be related to fundamental differences in their GABA to glutamate ratio and how their cortical mini columns developed. And therefore, it's possible, and some of them will tell you, that you are torturing them when you continue to expose them to one of their sensory over-responsive issues. And it suggests that the treatments that really emphasize coming up with sensory neutral environments, or helping them to, if they need to wear headphones in certain places, they need to do that. Whereas with our anxious kids, we'd say like, suck it up, we're doing this, we're going in, it's happening, right? These kids, it may not work. And then when we talk about sort of helping them, they still do need to deal with the world. When we talk about helping them sort of build their coping strategies, it might have more to do with distraction and techniques that help them handle that sensory information instead of sort of the way we think about it in the anxiety work. So I think that's something that we're still working on, but it's something to consider from your patients. We're working on, with this data, increasing our subject size, and I told you I'd come back to this. If you know any 15 to 18-year-old boys who are typically developing, I would like to put them in the scanner, please. Send them my way. Actually, up till 18, so 17.9 years old. I'm in particular need of those ones. So we're going to be able to compare across groups and do more with this data set. Going forward, and already Shula, Marella, and Susan are working on looking at youth with anxiety. And Shula is particularly interested in looking in youth with early life adversity and trauma to examine the relationship between sensory over-responsivity, GABA tone, and then neural connectivity measures to identify the common and distinct neurobiological mechanisms that may exist between these disorders. My very particular interest that I'm developing research in is studying youth with ASD and trauma or early life adversity to better characterize what PTSD looks like in ASD. I don't think we really know. And to examine the effect of these Salience Network alterations and SOR on how it affects the fear learning and social emotional processing in these kids at the behavioral neural level. And I think that will also tell us about how fear learning and exposure response therapy works more generally. I'd like to say thank you to Shula, Marilla, and Susan a lot, and then all the people from my lab. And thank you so much for listening. Do you have any questions? Oh, it has to be orchestrated, sorry. Tell me about it.
I'll ask the same question. Does anybody have any questions? Sure.
Have you done any longitudinal studies or are you planning them or is that what you were talking about at the very end, that you want to take normal folks and see whether these balances change with, because of course there's a lot of social anxiety in late adolescence, and I wondered if you could see shifts in your paradigm as you explain it to us.
Yes.
Over a longitudinal period.
Yes, so we do have some plans for that, and we're bringing kids back in who have previously gone through some of these same fMRI studies and some of the same behavioral work. Obviously, we're sort of adding on as we go, and so the GABA stuff is newer. We don't yet have sort of multiple time points on that, but it is part of what we're looking to do and part of what we're looking to get funding for.
Dr. Lee, the study that you mentioned last, I think the one that you were associated with, sounds like it's one of the few or only prospective double-blind studies that's been done. Was that accurate? I wasn't sure that you explicitly addressed that, but the related question is you used that one antidepressant treatment and whether other antidepressant treatments might have a different or similar effect on changes in the biome, whether that would illuminate whether it's really a cause-effect relationship or some other thing.
So I believe this is the first study of that type. I couldn't find any study like that in the literature. That the question that you have proposed is really what would be really exciting to see if there's any antidepressant dependent effect on microbiome. Is microbiome a mediator of antidepressant effect? Can you look at match the microbiome profile with the relationship with the antidepressant response so that you can identify who would be a responder to Lexapro versus venlafaxine versus Fatsima? I don't know, that would be a dream, but yeah.
First, these were both really excellent talks. I mean, I was really nice job. Emily, you alluded to the typically developing group, and I wonder if you can speak to the possible specificity of these neural effects as they relate to autism, say, versus anxiety or other disorders, and what those implications might have for treatment. We don't want to throw exposure away for everybody, right?
No, absolutely not. Absolutely not. So interestingly, in the first run of looking at the typically developing folks, they have actually a similar relationship between their GABA and glutamate ratios and the parts of the brain that the thalamus is firing more with. So their maps don't look a lot different from this, which suggests that more generally the GABA glutamate ratio is important for how this network fires, and that there might be another place we need to look to figure out what sort of the more specific differences between autism and typically developing controls for where the Salience Network is getting off track. But I think that we've seen there have been studies that have shown that when we do exposure response prevention therapy in sort of typically developing anxious kids, we can actually decrease their amygdala activity over time. And that seems to be a real thing that we definitely shouldn't throw the baby out with the bathwater. But I am suggesting that sometimes we have kids who are really telling us that they're suffering, and that we might need to start listening to them and really sorting out what that means for them. Is it something that maybe is really sensory based and their sensory system is actually different from yours? And is it something that's more about the anxiety about the experience? And I think that's important to think about.
Hi. So you showed these very interesting effects on the Salience Network with the decreased GABA inhibition. Have you found that decreased GABA inhibition relates to positive connectivity between the thalamus and sensory cortices?
There has been some evidence that there was actually a little bit of decrease in a couple areas, but no, in this case, we haven't seen a big difference.
Hi, thank you for the talks, and I just wanted to circle back to Dr. Piacentini's question about specificity in the sense that we see in individuals who are at clinical high risk for psychosis, I mean, interestingly, a very similar pattern of thalamic overconnectivity to sensory motor association regions that actually predicts, prior to onset, predicts conversion to psychosis. And the other interesting thing that I was curious whether you had looked at the hippocampus at all, because we do see this interesting kind of push-pull relationship. I mean, in terms of functional connectivity, I'm not sure about the spectroscopy aspects, but under connectivity of the hippocampus, but those same regions where we see thalamic overconnectivity. So I was curious if that was something you had looked at.
We have not yet. We're sort of waiting to do some final analyses until we have the full set, but that's very interesting. And I think that you're really getting at exactly the point of this work, is that it's interesting in autism, but it also seems, I think, different parts of this network we're going to find are issues in each of these disorders. And this having some sort of broadly less well-tuned connection between the thalamus and the cortex, which we're seeing in these disorders, is really important, and important for us to think about in terms of approaching these patients.
Thank you. I have one more question about the microbiome. So are we at the stage yet where we have essentially a standardized understanding of the nature of the microbiome in terms of the various organisms that constitute it, let's say in the stool, for age and sex and so on and so forth? Because how otherwise do we then interpret the sort of materials that you're presenting to us? Because if it's very difficult if it varies so much to understand where we're starting from, yes? And I was wondering if you had before and after studies again, and also whether there was any standardization across the sex and age spectrum as yet that we can rely upon.
So, as you mentioned, the sex. Oh, are you talking about the data set? Oh, so in general, sex definitely seems to be an important mediator, as we have seen in the association study with the probiotics. Only men, and the sex comes up as a mediator in other microbiome and mental health studies. It seemed to be important for my tiny pilot study as well. I don't know why we only had responders who were male and not female, but I think it is an important factor to look at, and you have to use, perhaps it might be, it might mean that there are different subtypes of depression. Right now the analysis, we just lump all depression as one entity, but it seems like there are certain type of depression that seems to be more sex mediated. So to answer your question, the large population cohort study that I presented, that one was a nice data set by using a really large set and co-varying, or being able to adjust for all the covariates, including age and sex in that case. And that was able to show consistent difference in specific taxa. So it is possible to interpret microbiome data, it seems, but you just need a really large data set, really large power sample size. Exactly. Variance is enormous, and there are several different ways to look at the microbiome composition. I didn't go into detail, but there's also a understanding that something called enterotype. So it isn't based on single genus. You don't give each genus of bacteria same importance, but you actually group them into subgroup of genotypes, or enterotypes, and then look at that as a marker of composition. And some studies have actually seen more interesting differences with the enterotype-based analysis. So it's very messy. Really, really smart bioinformaticists have dedicated a lot of effort into fine-tuning the analysis methodologies. I'm just keeping up with it is really difficult for me. Yeah.
I have another question thanks for your talks, guys. They were terrific. Another question for Melanie. So it's really interesting, the correlations between the different types of probiotics or different types of micro sort of organisms. But many of the ones that you presented data on aren't available commercially to people. I'm just wondering what you think in terms of treatment and moving forward relevance to, and like you mentioned your patients who ask you, should I take a probiotic? What do you say to them?
So the answer is, it can't hurt, but we don't have any data that supports any specific bacteria that actually shows strong evidence for improving health. That's the actual scientific data. That's what I was trying to refer to in the beginning, the separation of fact and the fad. And a lot more research is required to understand each individual variability, and what about their microbiome composition is a marker for who will be a responder to what type of depression or whatever the outcome may be. And it may mean that you may have to use different types of probiotic strains on different people. So we're talking about a huge black box here. So I'm just scratching the surface and just showing you that it is a very complex system. It is an important system. There seems to be some difference between pre and post treatment. There seems to be some difference between healthy and depressed. But in terms of how you use that information and translating into intervention, it is really preliminary. That's the conclusion.
Any other questions? Oh, sure.
So for Emily, can you fix the GABA-glutamate imbalance with GABAergic agents?
Probably not, because we've tried to do that by giving gabapentin, Neurontin, whatnot. So it's not quite that simple. But there are some new drugs coming on the market, actually, that interact at the GABA receptor in other ways that I think we should keep looking into. But at this point, it doesn't seem like you can just change the balance like that.
I have a comment about that. Actually, I saw in the literature that some of the function of microbiome is production of GABA. ASD and microbiome differences have been demonstrated. So there's dysbiosis associated with ASD.
Maybe you can't give them in a pill because their microbiome eats it.
All right. Well, thank you again very much for doing the talk.